So, a couple of quick things looking ahead to the next week in here, and then the next week in lab as well. So in here, after today, we have 3 more lectures before the next exam. They're all on, I'm just describing this as microbial communities. On Friday, we'll cover a topic that we were supposed to cover before the last exam, which is coming back to antibiotic resistance, but we're going to talk about it not from the cell standpoint per se. We're going to talk about it more as a social construct. So this was one where like some of folks had actually done the homework assignment ahead of time. I closed it for a while and I reopened it recently. So this is one where you have to watch a TED Talk and then give some thoughts in a group with you before coming to class. So take a look at that. And then on Monday and Wednesday next week, we will cover, Monday is going to be covering the gastrointestinal tract and talking about some resident bacteria of the gastrointestinal tract, and then talking about C. diff infections. Again, we'll talk about antibiotics here, but not in the form of resistance, rather in the form of effects on human microbiomes. And then on Wednesday, we will cover the microbiomes of the skin and the urogenital tract. And we'll take a look at a paper in particular that there's one group that's been investigating, actually numerous groups, but one in particular that's been investigating for children that are born by C-section or vaginal birth, how do their microbiomes differ from each other in the first month or so of their lives, and then starting to determine, does this have a long-term effect? Okay, and those studies aren't complete, so I just kind of want to give you, we're going to just take a look at what's been done so far. So that's what's coming down the pipeline, and then our last unit is kind of, I guess it's more health-based. We're talking about immunology. We've got about a week where you guys are going to actually present, and then the last 2 sections are epidemiology and vaccination, and that's it. So almost to the end. I can't believe it. Anyway. So that's the first part. Second part is tomorrow in lab. I posted an announcement. It went up this morning that just said to have for lab tomorrow. Your group and you should come with all of your stuff that you've gotten back from me so far, particularly the protocol and the guess my hypothesis sheet. What I have been doing is taking notes on each group on things that I want to make sure get addressed during our one-on-one meetings tomorrow in lab. These shouldn't take more than 15 to 30 minutes. I assume it's going to be somewhere in that time frame. And this is an opportunity for you to have my undivided attention to ask any questions about the project that you want to. It's my opportunity to just address any concerns that I have with where you're at right now and where I might see that there are particular lapses in the progress of the project to this point. Okay. Next week on Tuesday, we prep. Next week on Thursday, we begin running experiments. And by prepping, I mean, so for some folks, I know you need saline solution or you need sterile bottles. We're actually going to go into the autoclave room and use the lab as well to prep media, reagents, etc. So we have all of that ready by the time the next lab rolls around. At least by Wednesday, because I know some groups are bringing stuff in with them. Like Jill, I think you guys are probably collecting Thursday morning and then bring it with you to lab. So we'll make sure to have that stuff Wednesday so you can pick up, grab what you need, and then bring it back with you. Okay. Any questions on those items? All right. I forgot one last thing. So tomorrow, as you may remember, I said I was going to be gone in that 10 to 12 lab for a short period of time. Giving me probably enough time to meet with 2 groups during the lab period. I've already had one group say they'll meet with me at 9:30, which is perfect. That makes life easier. I'm still looking for one more. How viruses can be used as beneficial organisms. Oftentimes we think about them from a health perspective and think these are bad. If you've had flu, it sucks. If you've had mono, it sucks for 3 weeks. If you've had mumps, which I'm going to assume nobody in here has, it's not fun, okay? Viruses, typically we think of the downside of these. But there is an upside. And we kind of preluded this about a week ago, talking about the idea of phage therapy to block bacterial infections or to treat bacterial infections. And this is one example, for those of you that read this, this is one example where we've seen this being used within the past few years. And this isn't the only study, or this isn't the only time that it's been used. In fact, there's a group out in the UK that is looking at using bacteriophage to treat chronic ear infections in children. That would have been really helpful for Jake. We had to end up doing tubes in his ears because he would get one every single month, which would keep us up all night long because for a child, ear infections are painful and not fun. So there are groups looking at using this on a more broad scale, just other than just this condition here. But this is kind of a new thing to the United States. Okay? For those that read this, what's our, let's put this into, let's use this as a way to think about where we're going today. In the case study, we'll kind of walk through the investigation of the bacteriophage that was used. But what's the story here? So what does this article discuss? Deb?
So this guy had something wrong, and so we had to go into the hospital. He had a coronary bypass surgery or something where they put a plastic mesh in his
Yep.
That way it grew out of that and stuff like that.
Yeah, so one of the things to point out here is that they don't really know where the Pseudomonas came from. Could have been something on what we would think would be the sterile product that they've put into him. Could also be what we would call a nosocomial infection or a hospital-acquired infection. Maybe it came from another patient and then by improper practices somehow got transferred to him, or improper disinfection from the last time the room was used. Or it could have even been that this is just an infection that was caused by himself. In other words, Pseudomonas can be found on the skin. And so if the site where his surgery was performed and where he had the open wound wasn't being properly taken care of, that could have led to an infection as well. Okay, so the term we use for those types of infections are nosocomial. The other thing that I want to bring up is Pseudomonas is what we call an opportunistic pathogen. It's not one that normally causes disease, but if somebody is immunocompromised, in this case somebody who has just had surgery and a break in the skin counts as being immunocompromised here, that would put him and anybody else at risk of potential infection. Okay? What else happened? What else does this story talk about? Ashley. Yeah, so they try to treat it with a number of different antibiotics. One of the things that's really interesting about Pseudomonas is it's actually incredibly antibiotic resistant. And it's not just because of mutations and vertical gene transfer. It also can occur from horizontal gene transfer. It can also occur from actually a couple of features of Pseudomonas. So one structural feature we haven't talked about this semester, biofilms. Anybody know what a biofilm is? So a biofilm, essentially, the best example I can give is the plaque on your teeth.
biofilm is oftentimes secreted by microorganisms and creates a solid structure for them to, oftentimes they kind of have, once they've formed, they have the consistency almost of concrete. You'll find inside them, some of them can be more slimy than that. You'll find inside them bacteria. And this biofilm oftentimes provides resistance to antibiotics. You also start to find some diversification in there as well. And it creates this really unique microbial community. In a biofilm formed by Pseudomonas, you might find Pseudomonas in there, you might find Streptococcus in there, you might find Staphylococcus in there. So you find numerous different types of microbes, and there's an interplay. Oftentimes, in these biofilms, you find some sort of mutually beneficial relationship for all the microbes that are within there. The other thing is that Pseudomonas aeruginosa forms a really, it's oftentimes called a mucoid layer. Essentially, it's a slime layer. Anybody remember what a slime layer is, or another term we used for it? Ashley? Yeah, slime coat, and there's... It does, and it also prevents phagocytosis. So the other term we've used in the past, something that's related to the slime layer, is a capsule. The difference between the two of them are capsules are more bound to the cell oftentimes. The slime layer can be sloughed off pretty easily. But this prevents phagocytosis, helps the organism to attach to surfaces, colonize. The biofilm helps to colonize as well. And so what we find with Pseudomonas aeruginosa is it's so challenging to treat because it's protected by these different extracellular polysaccharides that it's secreting. Those also allow for it to colonize and remain in an area for a long period of time. So Pseudomonas is, actually we find it associated with some urinary tract infections in individuals who have catheters. The biofilms can colonize within the catheter. Eventually, bacteria move up the catheter, cause a urinary tract infection. Okay? So those are kind of the features here of Pseudomonas. What I want to do today, and we've got 35 minutes to work on this, is I want to work through a case study. And basically, this man's story began with a paper investigating the bacteriophage that they used to treat him. Ultimately, the treatment of his Pseudomonas infection with bacteriophage and with an antibiotic had essentially here a synergistic effect, one in which the two of them worked much better than either of them did individually. Long story short, just to kind of put this into context, the bacteriophage bound, what they found was it bound to a particular protein, OPRM, which is the one that you had to look at for the homework. Okay? So reviewing that real quick, in strains that, let me see if I can pull this up. All right, so in strains that have, or in wild type strains versus ones in which OPRM is deleted, what is the general trend for each of these antibiotics that was investigated? Haley. Okay, so the minimal inhibitory concentration for the wild type strain is much larger, and some people noted this could range anywhere from like twofold larger to, I think in some of the cases it's like, for carbenicillin, for example, it's like 128 times larger. Okay, what does that mean? So putting into the context of an interpretation here, larger minimal inhibitory concentrations, which of these organisms is most resistant, more resistant to antibiotics in general? The wild type. In other words, OPRM has a role in protecting these cells from antibiotics. It's an outer membrane protein. It is a protein that is bound by bacteriophage, since it's located on the outer surface, what might it be involved in? What might we assume that it's involved in to provide antibiotic resistance? Yeah, it's an efflux pump, most likely. And that's what was known before the investigation into this bacteriophage occurred. So keep that in mind, okay? And we'll walk through the story of how they identified essentially this bacteriophage and then the effects that it has overall on rendering Pseudomonas aeruginosa more sensitive to antibiotics, okay? What I want to do is I kind of want to do like a 10-minute, you guys work, 5 minutes, we'll talk. We'll do that a couple of times and at least get a couple of rounds in. We may not get all of this done. That is perfectly okay. But I want you to start to take a look at some of the data here to get an idea for what are these bacteriophage actually doing. So the first part, we've basically walked through the first part of the story. Just a couple of things to point out real quick. There are a number of different strains that were used in the study. So PA01, PA14 are lab-adapted strains. PAPS, PASC, and PAD-FU are clinical isolates. And then 1845 and 1607 are ones that were isolated from household sick. Right. I'll give you about 10 minutes to work on, there's an A and B on the front describing, reviewing the basics of lytic life cycle for bacteriophage, and then starting to take a look at the OPRM mutation itself, and if you can, get to the prediction of what you might think the growth would look like with and without bacteriophage added. Okay? All right, go ahead. And feel free to work with other people on this. It's a protein, that's a good start. What's lysozyme? Enzyme that breaks down bacterial cell wall. A lysine is the same thing. Okay, a lysine is an enzyme that breaks down the bacterial cell wall. Lysozyme is a specific example of a lysine. All right. So, in looking at this picture up here, each of these dots, and you may remember this from when we did it in lab a few weeks ago, each of these dots represents an individual bacteriophage that infected a bacterial cell and then lysed it, infected bacterial cells around that initial one, lysed them, so on and so forth. And that lysis that we observe kills the cell, leaving these blank spaces or holes or plaques where there is no bacteria. Okay? So when we consider that in the context of this next question here, which is, so they took OMK1, which is the particular bacteriophage strain that they're using, and then they tried to infect a strain of Pseudomonas that lacks OPRM. Okay? If OPRM is the receptor for the bacteriophage, okay, in cells that lack OPRM, would we expect infection to take place? No, because what is the first step of viral? So again, it will infect the wild type, but would not infect the mutant. What is the first step of infection, a viral life cycle? Attachment. So that attachment is absolutely necessary for infection to even take place. Without the OPRM gene, we wouldn't see infection occurring. And that's essentially what's happening here. So what I'm trying to show is you would pour a lawn of bacteria onto the plate, you would serial dilute your bacteriophage, and then just put small 5 microliter drops onto that lawn of bacteria. At higher concentrations on a wild-type strain of bacteria, you'd find complete lysis of all the cells where you've put those bacteriophage. And that's what this is representing. Eventually, you start to see individual plaques again, because the phage have been diluted out far enough that now only a few bacterial cells are dying. On the right there, we see a strain that lacks OPRM. these cells would be less sensitive or more resistant to infection by the bacteriophage. Therefore, it takes a higher concentration of our starting bacteriophage sample to even see infection occurring. Does that make some sense or connect some neuron transmitters? So when we look at bacteriophage infection, we can do these types of experiments where we can test what's the receptor. Wild type cells would be infected by the bacteriophage. Ones that lack the receptor gene, regardless of what it is, would be resistant to infection. Okay. Okay. So the next question is Channon Turner, the research scientist and the professor that were performing this work. Let's imagine now they do spectrophotometry to look at the growth of Pseudomonas aeruginosa. And the setup here is, just to kind of in part, this is a bit of a review of old terminology. 1 hour nope, that should say lag, damn it. 1 hour lag phase. OMKO was added after 3 hours of growth. And you have another sample that lacks OMK1 being added. So this is essentially asking, what would the growth curve normally look like, and then what would the growth curve look like once OMK1 has been added, given that that bacteriophage infects Pseudomonas aeruginosa? I'm going to give you a minute to think about this, but what I will do real quick is draw out kind of an example of the wild type cells. So if we've got a 1 hour lag, be something like that, then what do we have after lag phase? Log phase, so we see rapid growth, and eventually we reach stationary phase. And probably over this time we wouldn't see any death phase. It's not a long enough growth curve to see that. So let's say that this is roughly what we expect the growth of Pseudomonas aeruginosa to look like typically. What's it going to look like when phage are added? And I'll give you a minute to draw that out. So this one is just saying in the context of an infection of wild type cells, so of PaO1. So at 3 hours is when the phage are added. What do we expect to see then after 3 hours of growth? Probably, because we'd probably see death here because we know the bacteriophage is capable of infecting this wild type strain. So usually what we find is maybe early on kind of a low inhibition of growth and then a plummet. Something like that. Okay. That would be ideal. Any questions about what this growth curve looks like? And I think, Ashley, you hit the nail on the head. That's death phase. That's really what's happening there. Okay? All right. So with that said, I kind of mocked up what I thought these might look like, and it looks pretty similar to what we just looked at. What I want you to do is to follow up with the 2 questions afterwards. So and this is on this page, what did the phage do to the growth of the organism in the first few hours after it was added? And then there's a second part based on the curve that I've drawn that I want you to take a look at too, which is hypothesize why there might be regrowth of those bacteria cells. I don't know if that's enough. particular experiment, I mean, the beginning fits really well with what we'd expect. 3 hours, we see kind of maybe a loss of growth. Then we see a sharp decline in the amount of bacteria, concentration of them. But right around 11, 12 hours, there's regrowth. Why? Ah, bingo. Mutations. Mutation to what? Mutation to the bacteriophage, which would mean what is a potential gene here that was mutated? The efflux pump, OPRM. Exactly, and that's exactly what they observed. So they ended up testing, they took bacteria from this later time point, they ended up sequencing the OPRM gene, and what they found were single point mutations that led to most often missense mutations that inhibited the ability of the bacteriophage to attach to those bacterial cells. So therefore, they've now generated a population of Pseudomonas aeruginosa where that OPRM gene is mutated. Anybody want to take a guess as to what that means in the context of treatment of Pseudomonas infections? And think back to the homework. What effect would this mutation have? Meg?
So would it be really hard to try and give it like an antibiotic because it just regrows?
It does have to do with the antibiotics, but it wouldn't necessarily be harder to treat it. In the homework, what did we say about OPRM? Those cells are more resistant or sensitive to antibiotics? The ones with OPRM are more resistant. The ones where it was deleted were more sensitive, right? This is not a deletion, but it is a mutation. So we could make two hypotheses, really, right? One would be the mutations are going to make these harder to treat because there is a mutation to OPRM that actually makes it a better efflux pump. That's the term I'm trying to use. Or we could make an assumption that these cells are now actually more sensitive to antibiotics. The mutations that have occurred are ones where it disrupts the function of OPRM as an efflux pump. Okay? Everybody following along with that? Kaylin, did you have anything else to add? Ashley, anything else to add? Okay. I want to skip one of the questions on because we just kind of covered that one, but I want you to take a look at the table below. So what they ended up doing at this point was taking a look at the growth of, they divided their cells into two populations and tested individual isolates of the original strain that they were working with, or the strain of those regrowing cells, those that had become resistant to OMK01 or to the bacteriophage. And we can assume that these ones contain a mutation in in that OPRM, or that multi-drug resistance pump. This is a broth dilution experiment. That's a broth dilution experiment. So these are measurements of resistance to tetracycline in micrograms per mil for each of these different bacterial strains that they were working with. Again, these are the lab-generated strains. These two, 1845 and 1607, are those ones isolated from a household sink. These are clinical strains at the bottom here. Okay? Take a look at these results and describe what are you observing. Is one of those populations, the resistant or sensitive cells, more resistant or sensitive to antibiotics? I'll give you a couple of minutes. What's the trend? In general, what happens?
The OMK01 resistant has a lower enrollment concentration than the sensitive.
Perfect. So those that are resistant to OMK01, those that are resistant to the bacteriophage, and we can assume have a mutation in OPRM that has made them resistant. These are all now more sensitive to the antibiotic. It takes less of that antibiotic to inhibit their growth. Those ones that were sensitive to the bacteriophage in the beginning are now more resistant to antibiotics. So there is, one is, yeah, I'll just leave it at that. Exactly what you said, Derek. So in taking a look at this then, what does this mean in terms of clinical treatment?
You should use both. So.
Exactly, and that was the idea behind this, is let's use both. Let's not just use one, because as Ashley pointed out, what the researchers saw here, at least with this patient, was that he had recurring infections of Pseudomonas aeruginosa. They'd kill off most of the infection with some antibiotic. He'd be fine for about a couple of months, and then the infection would come roaring back. And so what some researchers are suggesting is that we need synergism here. We need to hit it with two different types of treatments. What we actually see here is a principle known as evolutionary trade-off. And what that term means is that as an organism becomes selected in an environment, as it gains some sort, and I put gain in loose terms here, but as it gains a mutation that makes it more successful in an environment, that mutation can have also a negative effect in a different environment. So how does that apply here? Well, in thinking about this example, those cells that are treated with bacteriophage gain resistance to that bacteriophage. But what do they lose?
The efflux.
They lose the function of the efflux pump, and therefore they lose their resistance to antibiotics, at least to tetracycline. Okay? This individual then, the way that they ended up treating him was supplying him with, and this was kind of like last resort. This is the kind of thing that earns you some sort of really big prize down the road, but went through no clinical trials before doing this. This was a very last resort thing. They basically walked up to him and said, how about this as a treatment? He signed a waiver saying, sure. They ended up doing it, cured, within a couple of weeks. The wound finally healed after 3 years of remaining open from constant Pseudomonas infections. So the point that I wanted, or the main point today that I wanted to make was viruses can be good. Okay? It's a matter of context. And in the context of clinical usage, there can be great benefit to using them. If you're worried about the mutations that bacteriophage can create to bacteria, here's one thing to point out real quick. For every single bacterial cell, and we think we have somewhere on the order of about 10 to the 30th on the face of the earth, there are 10 more bacteriophage. In other words, the number for bacteriophage is somewhere around 10 to the 31st. In other words, bacteriophage are always ahead in the evolutionary game relative to bacterial cells. At least that's what we think. The other principles that I wanted to make sure we covered today were just some things that we've talked about before. What is the effect of a bacteriophage on a bacterial cell? What does that actually look like when we visualize it? And then finally, re-hitting that idea of antibiotic resistance. Okay? I won't cover the rest of this, but I just kind of talked about the last page here, which is the evolutionary trade-off. If you want some more practice, what I would suggest is there's one more growth curve to take a look at in here, and for you to predict what would the results look like if OMK01 was grown with no tetracycline, which is the antibiotic they use in this part of the case study. What would it look like if it were grown with tetracycline? And then, no tetracycline with OMK01, no tetracycline with the bacteriophage, and then with tetracycline without bacteriophage, with tetracycline, and with bacteriophage. So taking a look at 4 conditions: no treatment,
treatment of one, treatment of the other, treatment of both, what would you expect to take place? Okay? And that just kind of reiterates the last point there. All right, with that, have a great rest of the day. I'll see you all on Monday. There is no mini lecture, it is the video, the TED Talk to watch. Friday, sorry, that's why all of you were looking at me like I was an idiot. No, I will definitely see you Friday.